in my lab studying one of the interesting complex called the axon junction complex. So my lab actually interested in the neurodevelopment and how associated with all kinds of neurological disease. So we all know actually the brain actually started with one single layer of the neuroepithelial cells. We actually also call this layer of cells as a neural stem cell. So this layer of the neural stem cells will undergo divisions, asymmetric division to proliferate, and also asymmetric division to produce neurons. And those neurons actually will migrate towards the outer layer to form the giant network in our brain. So it's a beautiful network. And my lab actually interested in how abnormal neurodevelopment will lead to the neurological disorders. So one particular protein we are interested in is actually the axon junction complex. So we all know actually the transcriptomes actually DNA, then it will transcript to the mRNA, but post-translational modification is actually very complicated. So actually we start with a pre-mRNA, then we'll undergo the splicing and the modification, export to the cytosol, then it will translate into protein. Eventually, the mRNA will be degraded by also very complicated process. So the axon junction complex include the RNA, EIL4A3, BTZ, and MAGO. This complex actually involves a step of this regulation. First, actually, it's bind to the axon junction and work together with the spliceosome to help the correct splicing. And then it's actually associated with the exporting and the importing will help mRNA to export into the cytosol. It will also interact with the translational machinery to stimulate translation. And eventually, this is actually have been well established. It's involved in the one of the evolutionary conserved degradation pathway called the nonsense-mediated mRNA decay. So how this actual protein involved in the mRNA degradation? So this pathway actually is actually interesting. It can detect any most of the nonsense mutation. So we know actually if the protein has the nonsense mutation, will produce the truncation or deletion. It will eventually impair the cellular function. So how does the axon junction complex help to detect this nonsense mutation? So we know actually if the mRNA will transcript to prime mRNA and splice into the mature mRNA, the axon junction will sit on the every single axon junction. And the first round, the ribosome will kick out all the most of the axon junction before it's reached the normal stop codon. So if there's any premature stop codon, what you can expect actually, the ribosome will hit first premature stop codon before the stop codon. So at that time, they still have the axon junction complex sitting in this mRNA. So the axon junction complex will interact with the ribosome at the first round translation, will initiate the degradation pathway. So this is how actually the axon junction complex can involve in the NMD pathway to protect cells from the nonsense mutations. So this is actually a very conserved pathway. And interesting, those proteins actually involve some of the disease. One of the disease we know is actually a mutation have been found in the recently published in the Nature Genetics. Is some mutation in the RNA genes cause this thrombocytopenia and the absent radius syndrome. So this is actually a rare syndrome, but the patient with this mutation normally develop the short arm of the furling development is abnormal. And also, they have the low platelet number, so it's called the thrombocytopenia. So you can see the mutation has the, normally have the one of the 1q21 deletion, big deletion, and some mutation in the other chromosome that actually lead to reduce of the RNA protein. So this basically is a haploinsufficiency or the recessive mutation. So this patients also actually associate with other abnormality. For example, they have defects in the kidney and also cardio abnormality and also mental retardation and abnormal brain development. So the first thing we look at is actually how this gene involved in neurodevelopment. So if you don't have any background on the neurodevelopment, here is actually how the early embryonic development happens. So in the early embryonic stage, the new stem cell will divide and also proliferate. Then those cells actually sometimes undergo the asymmetric division to produce new progenitor cells. So the neuron, immature neuron, will migrate along this track outwards and form this actually inside-out layer. So first, the cells, first layer produce cells in the inner layer, and then it's produced the outer layer. So this is the inside-out manner. Then the stem cell actually still reserved in the ventricular zone and subventricular zone keep dividing until the adulthood. So basically, we still have a residual neural stem cell in the adult brain. So first thing we look at is actually how the RNA expression during neurodevelopment. So we isolate the brain lysate from the early E9, the embryonic day 9, all the way to the adulthood. Actually, we see interesting patterns that actually RNA is highly expressed in the early embryonic stage and then decrease. So conversely, you can see the SOX2. This is actually the early embryonic stem cell marker. It's decreased correspondingly. So the neuronal marker TUJ1 increased. That means the cells actually start to differentiate into neurons. So also, we look at the staining pattern in the early embryonic brain at E16. We know that actually stem cell actually localized in the ventricular zone and subventricular zone, and then the neurons actually localized in the cortical place on the top part. So the neurons with co-stain with the RNA, this our interest mutation protein, and then the neural stem cell marker nesting. So you can see the RNA is highly enriched in the neural stem cell population, but actually decrease when they differentiate into neurons. So one question, so how does RNA, why does RNA actually express in neural progenitor cells? What's the function of the RNA in the neural progenitor cells? So we have one very unique technology. We can actually knock down and overexpress the plasmid into the embryonic stage called in utero electroporation. So basically, we can inject plasmid into the embryo brain in the ventricle area, then apply this small current to deliver DNA into the embryonic brain. We can put the embryo back to the mother, pregnant mother, and let them survive for whatever you want. Normally, we start with E14 and then sacrifice at E16, so basically 3 days. We can monitor the cell proliferation, differentiation, and migration. So here is basically one of the example. We can see we designed 2 shRNA knockdown RNA, and in the cells, we can detect the knockdown effects. And then we look at how the RNA knockdown affect the migration. You can see the cells in the control shRNA electroporated brain is distributed evenly in the 3 layers of the cortical plate, intermediate zone, and the subventricular zone. However, when the RNA knockdown, we can see depletion of the neural progenitor cell in the subventricular zone. So it showed the quantification here. And also, it's increased the population in the cortical plate. So this data suggests actually knockdown of RNA promotes the neural migration and deplete stem cell pool in the early embryonic stage. So we further look at how this actually happened. Actually, we found actually we used BRDU as a marker. So the BRDU actually will incorporate into DNA only at the S phase of the cell cycle. So this is actually, we can label the progenitor cells. When we look at the, so the green cells actually will indicate that the cells are knocked down RNA. So we quantify the BRDU percentage in those progenitor cells. We can see the BRDU incorporation is significantly reduced when the RNA is knocked down. Furthermore, we look at the differentiation, look at the neuronal marker, the TOJ1. We found that TOJ1 actually increased, positive cell increase. So we hypothesized that the RNA is actually critical to keep the balance between the neural stem cell and the neuronal cells. So it may be critical for the cell cycle regulation. To test this cell cycle regulation, we actually use a cell cycle exit index. Basically, we inject BRDU to the embryo and label the cells for 24 hours. And then we use the Ki67. This is actually cell cycle marker. Any cells still remain in the cell cycle will label with the Ki67. So any cell actually exists cell cycle will be Ki67 negative cells. So we basically measure the GFP positive cell and BRDU positive cell in the whole population at the beginning. And any cell actually exists cell cycle will be Ki67 negative cells. So when we look at the cell cycle exit index, we can see the RNA knockdown actually increase the cell cycle exit. So basically, the RNA is required to maintain the stem cell in the cell cycle. So conversely, we also look at the overexpression pattern. We see that actually overexpression stimulates the proliferation, stimulate BRDU incorporation, and suppress the neuronal differentiation. Consistently, we also see once we overexpress RNA, it's reduced the cell cycle exists. So basically, RNAs require the progenitor cell proliferation. So this is consistent. Furthermore, we actually make the condition knockout model of this gene. So we flux the internal straight exon of this RNA gene and can see once we infect the fibroblast cells with the CRE, we can completely knock out the RNA in the cells. And strikingly, when we cross this flux mice with the nesting CRE, that's actually the neural stem cell specific CRE line, it will wipe out, actually delete one copy of the RNA gene in the neural progenitor cells. What we see is actually you can see this is a smaller size, and also the length of the size, the mice is smaller, and also the weight is significantly reduced. And also you can see here, this is actually P17 mice. Let's see. Okay, they can move around normally. But actually the knockout mice is completely paralyzed. So they cannot move. Eventually it will die at postnatal P20, around P20. So when we look at the brain size, consistent with our data, actually the brain size is significantly reduced. You can see this is a very dramatic effect. This is only one copy of the gene, still have one copy in the brain. And the proliferation, we look at BRDU, you can see the BRDU labeled cells significantly reduced. And also, after we dissect out the P12 mice brain, you can see the dentagyrus dendrites in the wild-type mice, this dendrites of the knockout mice is severely reduced. The cortex also show the cortex thickness is reduced. You can see I talk about the different cell layer. The layer is completely screw up. We use the specific marker for the COX2 is actually labeled the layer 2-3 marker. You can see the behavior. And we also see this, although it's low, but actually we can still see it in the dendrite and the nucleus. Then we look at the behaviors. They are less anxious because normally the mice or the animal, they try to hide in the bushes, and also they try to avoid the center part because that's where the predator can catch them. However, we can see the overexpression in the center. And also we look at the elevated plasma, we see that actually they have reduced the number into the open arm, so it indicates some anxiety behavior. So to summarize this talk, actors actually cause different disease, different syndromes. So we actually don't know. Actually, one aspect is actually they have particular or specific function in different tissues. That's what I can imagine. So maybe they interact with a different factor in the tissue and control different substrate of the transcripts in different tissue. Because although they are essential, but the expression, you can see the expression level is different even within the brain, right? So they basically, they probably have different function in the neurons, different function in the neuroprogenitor cells, different function in the macrophage. It's highly expressed in the blood system, so that's why the deletion actually caused the low platelet number. So
you think that tissue specific difference probably come from cofactors?
Cofactor or specific substrate. So they
expression difference.
Yeah, they actually, when we also did some of the RIP assay, it didn't bind every single transcript. So although it's they call exon junction complex, but actually it's not bind every single transcript. And some of the transcripts only have one exon, so it will not bind those. So it's not substrate. Okay?
Well, I think in the interest of time, we need to move on. So any other questions, please take offline. Let's thank our speaker again, please.
Thank you.
Our second speaker of the morning is
First of all, I would like to thank the organizers for giving me the opportunity to speak at this conference. Earlier, we put forward the hypothesis on the existence of a of a novel regulatory network, which we called transcriptional interference network. So this hypothesis is based on the interaction of the neighboring genes through the collision of their transcriptional machineries and the transcriptionally overlapping regions. So we proposed that this kind of interactions form a system-level self-regulatory network and result in a strictly controlled progression of the on and off states of the genes. We used the herpes virus model for the for these investigations to show that our hypothesis works in nature. So we originally assumed or proposed 3 different basic types of interactions. One of them is called as a waterfall model, which is represents a unidirectional interactions. Typically, 2 parallel overlapping genes can exhibit this kind of interaction, which means that the transcriptional machineries or the RNA polymerase of the upstream gene will somehow interact with the downstream gene. This interaction is typically the dislocation of the transcriptional initiation complex. The second basic type of interactions we called as a seesaw model, which is a bidirectional interaction. It means that both of the interacting partners exert an effect on the other genes. Typically, this occurs in convergently oriented genes. And we suppose that there is a third type of interaction, which we call as promoter competition model, which means that 2 divergently oriented genes overlap at their promoter regions, typically at the Tata boxes. Well, the thing is that our data refuse this hypothesis because this is not a competition. This is a joint control of the transcription, but I will show it a little bit later. So here is a complete set of the, let's say, subtypes of these interactions. I will go into details later when I discuss these cases individually. I only mention one thing before, that we can distinguish between the so-called hard and the soft interactions overlap. In the case of the hard overlap, there is a, let's say, a collision or interaction in any case. But in the case of soft overlap, there is an alternative. Normally, this is represented by alternative transcription initiation and transcription termination. So in one case, there is an interaction, and in another case, there is no interaction. That's why we will call it soft. The idea of the transcriptional interference is not a brand new idea. There were sporadic cases for pairwise interactions between viruses, bacteria, fungi, insects, and mammalian animals. What we claim is that this kind of interaction is not just a curiosity, but represents a widespread mechanism, not only in the viruses, but in the whole living organisms. So our model, what we use is an alpha herpes virus. This is a neurotropic herpes virus called pseudorabies virus. Pseudorabies virus is a popular herpes virus for the investigation of the pathomechanism of herpes viruses. It is also a widespread tool for the transneural tract tracing of neural circuits, and also as a vector for delivering foreign genes into neurons or cardiovascular tissues, etc., etc. So we use this virus for showing that the transcription interference is a really global mechanism. First of all, we sequenced this virus using the PacBio RS2 platform. And I'll show you a typical genomic organization of this virus. So there are genes in a tandem orientation, which is opposed with a similar cluster of the genes. And this genetic module is surrounded by divergent genes. And we can find every kind of interaction, every kind of overlappings between these genes. So this kind of genetic architecture calls for an explanation. That's why we try to investigate this issue. So first we determine the transcriptome of the virus using Illumina sequencing, both Illumina sequencing and the PacBio RS2 platform. And especially with this latter technique, we could detect several new transcripts, which was never been determined before. And this was allowed by the special properties of the PacBio sequencing. So we could detect very long transcript, typically polycistronic transcript. So in this virus, there are many different kinds of polycistronic transcription unit, but as it is known, the prokaryotic organisms contain this kind of units. Here, this is a little bit different because only the most upstream genes are very likely translated. The others do nothing. And we also could detect, for example, spliced and alternatively spliced transcripts, etc., etc. So let's start with the discussion of transcription overlap of the tandem genes. So actually, there are two different kinds of overlap. One of them is a so-called tail-to-head overlap, and practically all of the genes standing in the parallel orientation overlap in this way. The second type of overlap is, we call it as multiple parallel overlap, when the transcript share a common poly-A signals and also poly-A tails. Okay, and we can, I can show you here that the second case is most frequent here. So here are the polycistronic RNA, and the common poly-A has a largest peak. Here you can see that case. But we have shown that almost all transcriptional units have their own poly-A signals and poly-A tails. Many of them are represented only in a very few copies. Or there are also other genes. You can see here polycistronic genes, which can be transcribed individually, having their own poly-A tails. This was sequenced using a poly-A-Seq platform. So here I can show you the poly-A signals of the upstream genes. There are the major poly-A signals indicated by red or a blue arrow, depending on the orientation. And also there are rare, weak poly-A signals. The very weak poly-A signals are not indicated here. So I can show you the transcriptional dynamics of the virus. I will show you only some examples. For example, here we can see a cluster of genes. These are the USA 8, 9, and US2 genes. As you can see here, every combination occurs during the transcription. So the US2 genes can be transcribed individually. This is the same for the US9 and USA. And bicistronic and tricistronic products are also produced. So there's a special kind of dynamics. At the first hour, as you can see, the US8, US9 bicistronic transcripts are produced in a large amount, and the US2 in a smaller amount. Later on, at 4 hours, the US9 appears, and the proportion between the transcripts will be changed. The US2 will be transcribed in the highest amount. And this tendency is reversed once and then reversed back. So there is a special kind of transcriptional dynamics. So what we claimed, that the transcriptions interfere with each other. So it means when one transcript is, when one gene is very active, transcriptionally active, then it will suppress the other neighboring genes in every kind of orientation, the convergent genes, the divergent genes, and also the tandem genes. So when we analyze the transcript, and this is allowed by using the PacBio platform, because using PacBio, we can discriminate between the different transcripts, including the polycistronic and monocistronic genes. We can see a special tendency when one gene is expressed. I mean, this is the, well, this is a value, as R delta value. This is actually, there is the sequencing reads. It is called Roy reads in the PacBio sequencing. And we use the delta values, which means that we did use the R values at time t from the t minus 1 values. And we obtained this kind of data that there is an inverse relationship between the expression. So this is the aim of our work, to find a relationship between the expressions. So here we can see the so-called hard overlaps, the hard convergent overlaps. You can see here this case, the two opposing genes means that. So this is exemplified with the case of the U30 and 31. So the heads of the two genes overlap with each other, and you can see here that there are the messenger RNAs and also antisense RNAs. And there are poly-A signals for both of the genes. We showed that the transcription does not stop at the poly-A signals, but with a certain efficiency, there is a transcriptional read-through. And later on, both the sense and the antisense DNA strand is active. So what could be the explanation for that? What is the role for these antisense RNAs? This is the basic questions. So if we examine the transcription using real-time RT-PCR, then we also find an inverse relationship. So if the messenger RNA, has a tendency up, then there will be a reverse tendency, and vice versa. So this is the analysis using a real-time PCR. We can repeat it using the PacBio platform, and we obtain the same. So we can see an almost completely inverse pattern of expression between the two convergent genes. You can see that the Pearson correlation coefficient is minus 0.55. This is very good inverse correlation, but not perfect. This is because we should examine, the transcriptional activity, but we only can examine the transcripts themselves. The transcript has a certain, half-lives. Maybe at a certain times, there's no transcriptional activity at all, but there are transcripts from the earlier times remained in the cell. So we also can examine the effect of the joint effect of the tandem. Let's consider the UL31 genes. So there are two effects on these genes. One of them is its tandem partner, the UL32, and also the convergent partner. We can join their effect, and then if you see this picture, you can see also an inverse correlation. So if these two genes are active, then the middle one is repressed, and vice versa. The reverse is true if the UL31 starts to be active. So this is what I mentioned at the beginning of my lecture, that we supposed that there's a competition between the promoters at the overlapping regions. What we obtained, that in this situation, there's not a competition, because if we consider the UL30 genes and compare the expression with the UL29, then we will have a same expression pattern. So it means that if there is a Tata box overlap, then this represents not a competition, but a joint coordinated effect. So we have a hypothesis. Two major points was proved to be good, the third one not. Okay, then we can also see such kind of, this is only just an example. Here you can see that UL9 and UL8 genes, if one of the genes are active, then the other genes are not so active, and then there's a change in the expression pattern. Well, you can see, for example, an alternative. We can see that there are alternative transcription terminations. For example, this is the US31, US35, US36. Normally, so these genes stop be transcribed here, but in some cases and at some time points, there is a termination which overlaps with the US36 genes. Okay, thank you. So here you can see that there are convergent overlaps in this indicated areas. Well, this pictures was prepared 2 months ago. Today, we determined more interactions, more overlaps. So soft overlap, again, we can skip this, or maybe not. So I mentioned that with the case of the UL30 and UL31, there's a hard overlap. But the transcription continues across the polyase signals, and it produces antisense RNAs at the complementary DNA strand. This is the soft form of the interactions. If we examine this case with real-time PCR this is luminous sequencing, then we can see that there are antisense RNAs with a relatively large amount. These antisense RNAs are produced by the transcriptional read-through mechanism. Finished. Okay, then I would like to thank your attention, and this is the acknowledgement pass. This is my groups who are here in Orlando. I would like to thank to the UD Genomed and Mark Snyder at the Stanford University Center. Thank you very much again for your
Thank you. We have time for a very, very short question.
Sure, we got time.
Okay.
Any big questions?
All right. Thank you. Thank you. Let's thank our speaker.
Hi, yes. Very intriguing talk. Thank you. Since you can go ahead and crunch the promoter overlaps and transcriptional direction for genes in a known reference, and we have lots of transcriptomics data for those references, what would explain expression for genes that are transcriptionally close structurally that don't fit this hypothesis, where transcription of one hinders another and vice versa, but in real transcriptomics expression data, we don't see that kind of inhibition or consequential impact.
So the question is, if I could understand well, what is the mechanism for the interaction?
Well, if we see in observed data that there's plenty of now,
yes,
we don't see consequential impact of expression for one gene on another.
Yes.
When there's
So
overlapping or intersecting promoters or they're close structurally. What is then the explanation of your hypothesis with respect to that observation? If we don't see that inhibition, and yet they're structurally, physically close, or they have intersecting promoters, or one by your hypothesis would hinder another, then what if we don't see that?
So you don't. So we have a large set of data. We could not examine all of the data now, but those examples what we have examined, we could find the relationships what we expected, at least in the case of the convergent and the parallel reoriented genes, and we could not find the relationship between the tether-box interaction. But if the overlap is larger, because in this virus there are 13 divergent genes, and only 2 of them exhibit tether-box overlap, the remaining 11 exhibit a larger extent of overlap, and these genes show the inverse pattern. But what we show only the relationship between the gene expression. We did not show any direct evidence how this interaction occur.
Okay.
What's the question?
Perhaps we should take the discussion offline then. Okay.
Okay.
Well, let's thank our speaker again, please.
Some data about determination of the cause of male infertility using transcriptome. But nowadays, more and more couples have problem to get child. So how we can define infertility? Infertility we can define as the lack of conception following of frequent unprotected sexual intercourse during 1 year if the couple is under 35 years old, and then is older, and this is only 6 months. And approximately affects 15% of reproductive couples, and the ratio is increasing with the age. And what is important, men and women are equally also affected. The internal causes of male infertility are alcohol, drugs, tobacco, some health problems, too much radiation or chemotherapy, and also some environmental factors like too much pesticides, hormones in meal or lead, and also high temperature. In diagnosis, the most of male infertility causes are due to low sperm counts or poor sperm quality. The remaining causes of male infertility can be anatomical problems, hormonal imbalances, or genetic defects. In this year, European Association of Urology showed that 30 to 30% of male infertility is still idiopathic. So determination of the genes which are involved in spermatogenesis control might help to better understand molecular pathways driving male gamete production, and consequently the reasons of male infertility. In our institute, during several years, there were created and analyzed knockout mice models for several genes which were essential for spermatogenesis and then for reproduction. But we detected also several non-essential genes, and we work more with these genes. These genes are specifically expressed only in germ cells during spermatogenesis. It is histone cluster 1, which replaces histone in the first spermatocyte stage. Testicular haploid express gene, which is regulated from some factors from Sertoli cells. CAMP responsive element binding protein 3 like 4, which is active in response to the stress condition. And transition protein 2 is also replacement of histones during remodeling the spermatids. And testis express protein 22 is responsible for biogenesis of acrosome and midpiece in the last step of spermatogenesis. And last gene, acrosin propeptide, which is important for good acrosome construction, and then during fertilization and acrosome process. In our institute, all these genes were knockouted, but in single knockout, like double, triple, and more knockouts in different combination were every time fertile. But when we knockouted 6 genes, in some animals where we observed infertility, and it was not problem because we have knockouted all the 6 genes, but it was some other problems, and it was our question how only some % of these animals are infertile. So we analyzed the phenotype of the 6 times knockouted mice, mice, and determinate the infertility causes with also analyzes new genes which we obtained from transcriptome. So the first shortly about the phenotype which we received. We observed that about 20% males are infertile. In this time we also in these males we observed all stages of spermatogenesis. So spermatozoa were produced standard like normal, because it's known that serum testosterone level also influence on fertility. We checked the serum testosterone level and also total sperm number, and was similar like in this fertile knockouted mice and infertile wild-type mice. But we observed that the sperm speed from these infertile males and also acrosome reaction is destroyed, and also we observed increased number of sperm with abnormal head. Here you can see the how looks like the normal, and in different way destruction the normal shape of head. So because it's known that the transcriptome tool we can observe, we can obtain some clusters of co-regulated genes to the process, clusters of genes involved in the same biological process, or cluster of genes expressed in the same organelle. We decided to compare transcriptome, these fertile and infertile males. We observe, because it was also from literature known that about 2,700 novel transcripts were identified from these mouse germ cells like cytochrome C, which regulate apoptosis in germ cells, or another factors which regulate, which are transcriptor regulator during spermatogenesis. We create, we use a gene chip mouse array from Affymetrix. It was to some small plates, and we compared this fertile and infertile knockout mice. On this plate we tested 21,000 genes, and we obtained 55 genes which were down upregulated between these fertile and infertile males, and from them 41 genes have known expression in testes. 4 were testis specific. 4 genes were completely unknown. There was only short sequence obtained from some computational predicted database, and 10 genes which were known from other organs than testes. But for most of them, the expression in testes were not tested. So the first we tested these 10 genes, but only one of them was in testes specific only. But the most interesting was this gene. It was predicted gene, it was short gene, which was the strongest down-regulated between these fertile and infertile males. So we decided to analyze more detail this gene. When we confirmed this transcriptome via real-time PCR, we analyzed this gene, and in different infertile 6 times knockout males, the expression was not present, whereas in fertile was strong present. And also by quantitative PCR, we didn't see this expression in infertile animals. Then we analyzed the expression of this gene in different organelles, and was expressed in testes, and epididymis was really strong reduced, and another organs was not expressed. So we concluded that it might be interesting gene for fertility. Then we analyzed the expression of gene in different days postpartum in testes, and expression start in 16 days postpartum when primary spermatocytes are produced. This we analyze also in different cDNA obtained from mutated males mouse with mutation in with spermatogenesis arrested in spermatocyte, first primary spermatocyte, secondary spermatocyte, and spermatids, and were present. So these results confirm our results before. It was not expressed in testes and epididymis in mice with completely arrested spermatogenesis, and was also not expressed in Leydig and Sertoli cells. So we conclude that this gene is strictly expressed in germ cells. To be sure that this gene is expressed also in spermatozoa, we isolate sperms after mating, male and female, from vaginal plaque positive female, and we isolate and obtain this gene. As a control, in uterus it was not expressed. Also epididymis in 20 days postpartum was also not active. We also prepare castration of these males, and 3 weeks after castration we couldn't observe any expression in epididymis. So it suggests that this RNA might be stored in spermatozoa and useful. It's known from literature that mRNA might play a certain role in fertilization and embryonic development during different reorganization of DNA, formation of pronuclei, or activation of oocyte. It's also known from establishing the imprinting in early embryos. There is also some iRNA which inhibit genes active exclusively during embryonic development. And then we wanted to know if it's important to fertilization and developing this fertilized oocyte during early embryogenesis. We mated males with superovulated females, and 48 hours later after this mating, we checked the two-cell stage. This is the first embryonic step, and we observed that from fertile 6 times knockout, we observed this two-cell stage in 35%, but from infertile it was nothing. So the next question was it's active. Is it possible to fertilize or not? So to find the best time for isolation to pronuclease stage from fertilized oocyte, we try in different time points after fertilization, and from wild type, every time we receive about 20% to pronuclease stage, but from fertile it was every time about 3–5%. And in the last this last hours we received also 2 cell stage already. So it was not possible to establish good time to find this 2 pronuclei from potentially find this 2 pronuclei stage from infertile males. So we conclude that this time depends on fertilization time and takes place during 48 hours after mating. You never know when when directly the mating will be, the contact will be done. So to answer our question, we create some in vitro fertilization experiment. And the first, it was in vitro experiment with oocyte, where we used oocyte with zona pellucida, with complete oocyte. And here we observed a reduction from between fertile and wild type in 2 cells stage. So in infertile we didn't observe this 2 cell stage. So then we destroyed the zona pellucida using thyroid acid, and we could observe the similar percent of 2 pronuclei and 1 cell stage in all 3 groups: wild type, 6 times knockout fertile, and infertile males. So according to all our in vitro experimentation, it seems to be that the sperm from infertile males are not able to penetrate the zona pellucida to intact oocyte. So then we concentrated on this gene again and check and compare the sequence of protein with database. We find that this gene encodes a protein which is similar to capping protein alpha subunit family, and members of this protein family regulate growth of the actin filaments by capping the barbed end and growing actin filaments. In normal cells we have actin cytoskeleton, and this capping protein blocks polymerization and depolymerization this actin. So, and as we know, the actin is important to create the cell shape. It's important also in cell division, and the polymerization to allow the activation of the outer acrosomal membrane during acrosomal reaction, and prevents the sperm from incorporation into oocyte cytoplasm. It's also important in maturation of sperm and can block sperm motility. We also we also create antibody against a specific epitope in this protein, and this we observed expression. We observed the protein in testes, and then we also prepare experiment of colocalization this protein with actin capping protein subunit alpha antibody, and this antibody, as you can see here, it's nearly yeah, it's nearly 100% colocalization with this new gene, new protein. So we suggest that our gene might be a new protein, might encode new protein, which is new capping protein. So our conclusion, what might be the reason of infertility? We suggest that dysregulation of germ cells acting in cytoskeleton reorganization might be the cause of infertility in these males with 6 knockouted genes, because this gene is the protein encoded by this gene might be important for the regulation of the actin cytoskeleton. We observe increased number of abnormal head. We observe also reduced acrosome reaction and reduced sperm of reduced speed of sperm, and the problems with fertilization. Finally, I want to say thank you for Professor Wolfgang Engel, the head of Institute of Human Genetics in Göttingen, and also to all my colleagues from this university. So thank you for your attention.
One question.
One more, just a very short question.
Anybody have a question? Okay. I do believe we have a workshop too. That was kind of in the thing there, right?
Yeah.
So we'll go ahead. I guess no other questions?
No more
Okay.
questions. Okay. Let's continue for the workshop. Okay. So our next speaker will be Dr. Shangming Wang for the workshop.
Okay, also folks, just to let you know, the next session after break, we have 2 cancellations. They're not here. So we're going to be moving people up, okay? Again, I know a lot of people weren't here earlier this morning. When I'm back here going like this, that means you have 5 minutes left for your presentation and questions. Just please, I politely request that you try to comply with that so we stay on time and don't stay till 6 o'clock like we did. All right, folks, thank you so much.
Okay, Dr. Wang finished his master. Sorry. In Shandong Medical University in China, and then he worked as a assistant professor in Northwestern University. Now he's actually an associate professor at the Department of Genetics, Cell Biology, Anatomy, University in Nebraska Medical Center.
And you can use and then this is your laser. And give me a moment and I'll start recording, okay?
So basically, I'm talking about the same topic as I talked yesterday, but in a more relaxed time. Everyone knows gene expression is on the title regulation, but the actual picture is there's not much knowledge when we talk about any particular mutation with disease, because most of focus are in the coding region. Everyone talk about the mutation. It's coding some protein truncated, stopped, blah, blah, blah. How about the regulator area? It's really a serious challenge. As I said yesterday, current technology by no means provide a de novo determination for any genetic disorder in the regulator area. We really need to have some very powerful genome-wide tool to address this issue. And from our preliminary data work on breast cancer, I strongly believe the regulated area contain a rich information to address basic biological issue and also medical issue, and particularly in the cancer. No matter how much effort people make nowadays on cancer genetics, cancer genomics, not much about regulated area. That's highly unlikely. And in fact, when we talk about the cancer in general. We talk about the gene misfunction cause cancer. You look at all genome-wide study. Most of them are not very as expected. Say the large-scale cancer genome study. It really require use the sample size of tens of thousands in order to identify some mutation in the coding area with statistic significant. And we know every cancer, now they talk about personal medicine, talk about the precision medicine. You don't expect that so many things happen in random, say a genome, in somatic genome in cancer. I don't think there's any high chance to identify as a very common cause for a particular type of cancer. That's already proved by all genome study. Whenever identify some common mutation in the coding area, normally you see very low percentage of cases contain the same mutation. You not talk about the same position in the coding area. The thing about the coding gene only account for 1% of the genome. 99% of the genome are not coding for anything, but a part of them are regulated area, say about 2-3%. Even this 2-3%, there's no systematic analysis so far. Why? The tool. Really, there's no tool. That's why I'm suggesting, should we focus on more in the regulated area for particular disease study? Yeah, and I ask for your comments. Anyone make comments for this? What do you think about it? That's the whole point about the workshop, right? And that professor, yesterday you talked about the polymer, use polymer mapping promoter regulated area. Do you have any comments for that? Can push your system become a universal general tool for anyone interested in analyze regulated area?
Depends on how much data you want to look at. I mean, the problem with doing database searches is you get a large number of positives. And so you end up in database searching, constantly setting windows on, it has to be this significance or I won't look at it. I have to have sequences much or I won't look at it. But there's a huge amount of data there.
Your system can work on 384 ones, right?
Yeah, that's how many wells are on plate.
Will it be possible to push go to like array system with genome-wide coverage?
Yes, it would. I mean, essentially, well, the other limit to it is how much, essentially the density phenomenon. I was discussing this with a colleague yesterday, that you have to have it's about 10,000 ions before you can detect it in a mass spectrometer. And so you have to have a spot of a size that would contain 10,000 ions, okay? And so you could make a large array, you know, maybe spot 20 nanoliters instead of spotting 2 microliters like we did. And, you know, as long as there's 10,000 ions there, you would detect it, and you would be able to do analysis on it.
So this is a radical possibility. Practically, can you push go that direction?
Well, graduate students are only in the lab for 4 years.
Can ask Bioinformatics get involved, adapt to their system, high spot system?
Well, there are other methods than mine. I mean, for example, CELDI, which is where you covalently attach a DNA to a surface. Okay, that essentially in that system, the problem is you can have a very dense probe, which for DNA, DNA binds anything if you have a high enough amount, right? I mean, transcription factors bind to DNA with nanomolar affinity, but when you put a huge amount of DNA on a two-dimensional surface, concentration's undefined, right? It's just concentration's three-dimensional. How do you define it for 2 dimensions?
You guys think about whole genome sequence is a way to go to analyze a regulated area? Now it's much cheaper, and the lowest one whole genome sequence is about $1,300 whole genome.
Yeah, the biggest problem there is the quality of software for identifying binding sites, which was a point I made in a question earlier. I mean, we ended up doing one project that spent about 2 years where we were looking at a promoter it's involved in HIV, and the computer insisted there was an NF-kappa B site there. There is no NF-kappa B that binds to that DNA. So a lot of what you get out of a computer, it's kind of like doing a prosite search and believing that everything is myristylated or everything's phosphorylated. Computers predict things that aren't real.
Yeah, but now the DNA sequencing technology, particularly for next-gen sequencing, obviously the first lines create a huge amount of digital information. How to mine the data, get the value from the massive data set. Obviously, computer is essential tool. Without the computer, without bioinformatics, when rare cannot do anything.
Yeah, it's kind of like the old saying they used to have with computers, garbage in, garbage out, right? I mean, you essentially get what you're looking for in computers.
Yeah.
And you have to be aware of that. That's the only caveat, okay? Computers are wonderful tools. I certainly couldn't do proteomics without them. But the thing is, if it tells me that I have actin in a sample and the sample should not have actin in it, I tend to go with my own beliefs. I'll probably go ahead and look for actin, get an antibody and do a Western blot, but I'm pretty sure the computer is telling me something that's not accurate.
Yeah. And other people comments about that?
The thing about promoters, though, is you already know enough to be able to identify a large number of them, right? I mean, there are 23,000 genes, so there must be minimally 23,000 promoters.
Far more than this.
Oh, there would be more, because there are a lot of transcripts that have alternate promoter usage.
Yeah.
And promoters can occur in introns as well as in exons. So there's 23,000 minimum. But you also know a lot of characteristics of promoters. They have TATA boxes, they have INRs, they have DPE sequences, BRE sequences, right? So you do know what a promoter looks like. And I think by combining all of those things that you know, right? I mean, if you look at an intron and you find a TATA box and a BRE sequence, I'd consider that a possible promoter.
Yeah, but for promoter, for TATA box, if the gene has TATA box, we'll suggest this is promoter. But any TATA box sequence, if it looks like TATA box, is located nowhere in the middle of the intergenic region. There's no indication those are TATA box. That's a trivial part for computational prediction. They always pick up a lot of junk. They say this is TATA box. But in fact, that's nothing to do. It's just by chance with TAA.
Yeah, that's why I'm saying it has to be combinatorial, right? I mean, TATA boxes, It's very rare you ever see a TAATA for a Tata box. They don't look like that. Quite often, that second T is a C, for example, in Tata boxes, okay? And INR sequences are not completely defined either. I mean, they begin with an AG, essentially. Other than that, they're quite variable, okay? Same thing with Breeze. I mean, Breeze sequences are not clearly identified all the time, but if a computer can find many of those things, I would say that is a good candidate for further investigation.
Mm.
Can you say something about how variation in promote structure affect a real specific expression? A real specific expression. Now you see we have non-range sequencing. We can get haplotype. This is no problem. So now my question is, you see, you study the variation in promote area, promote structure. How variation in promote structure will affect a real specific expression. So I observe, you see, a real specific expression. The also, you see, there we can observe variation across the gene. Different genomic position, we observe different real specific expression. I don't understand. It should be the same, right?
Well, if my understanding, if in the regular area, particularly for some particular transcription binding sites motif, if there's any significant change for some highly conserved base, theoretically this should affect the expression level, either increase or decrease. And then this is, in fact, is theoretically same as the basic fundamental biology quantitative expression, right? Dosage effects. And the thing about the network issue, every gene expressed not only as yes or no, but how much. If the quantitative feature change, one of the key gene, if there's downstream 10 targets, that's obviously affect all network combination.
I mean, you see, a near specific, you see the same genomic position, two different area, you will have different expression.
You mean different organ, different tissue?
A real
okay.
specific expression.
Yeah.
Okay, and then there are not two questions. Question one, you see, you observe a real different area, we have different expression on the same genomic position. Second, different genomic position. Now, we have different area specific expression. Now, someone is he just assume on the same haplotype, these should have the same expression level. However, you see, I look at the notable data. I found, you see, there are no common expression level for the area specific expression. Now, maybe someone saw that this is caused by sequencing technology variation.
Well, that's a technical
However,
issue.
now we have long-range sequencing. We can sequence haplotype. Now just shotgun sequencing. So maybe now someone do some experiments, examine. So I don't know, gene negation. Many say I don't believe. Then my question is, I should observe data. You should explain the data. You not just the hypothesis. You're a biologist. Your hypothesis we should examine by experiments.
Well, first of all, there's a technical limitation. I think if there's 20,000 gene, you see 100 gene promoter variation. the benchwork can only adjust one by one. That's a much simplified system. Theoretically, I was thinking this is a network. This 100 gene affect the 20,000 network. And experimentally, I don't think it can prove this at this stage on the technical limitation. On the other side, theoretical biologists or computational biologists, they can model in this system. You saw a network, beautiful network picture, but neither, I don't believe it. How to prove this, right? That's the caveat of the current science. Biology is limited. Very traditional way, one gene, one function, one story. In the real field, in the real world, it doesn't exist. On the other side, the theoretical biologists always model something, which is not based on experimental data, because there's no data. And then use existing theory, Bayesian theory, for example. But this can explain everything. Like particularly for transcription factor mutation network, you can see easily nowadays, Nature paper, Science paper. Look, the picture are so nice, like artwork. I don't believe it, because there's no way to prove this. But this is current art of the science. Anybody have comments about that?
But a lot of times what those pictures are, aren't they, is a dramatic way of showing a hypothesis. And perhaps that is the complaint you're making, that it's hypothetical, that there's no data yet to support that, right?
Yeah.
I mean, we all have to do things like table of contents, illustrations, that we're trying to make one picture to say what we were doing, right? I just did one for a paper on the method I talked about yesterday. I essentially showed a promoter and an open reading frame, transcription factors binding, and underneath it I put question marks for where they bind, right? I mean, you know, does that represent the experiment? Yes, it represents the experiment. But if somebody takes that too literally, then they're going to be misled. They're going to think transcription factors are little color balloons and question marks exist in DNA, right? So we're trying to make a hypothesis that we can test. That's all the pictures on covers are.
But my thought is, in many situations, there's no way use current benchwork style to prove this kind of highly theoretical model. Very hard to prove this.
Yeah, I guess a lot of times we just stumble on things. I mean, like I've worked on CEBP for a very long time, and Dr. Liu informed me yesterday that it can be a tumor suppressor, right? That 3' untranslated region. And I doubt seriously that he went into those studies expecting to find a tumor suppressor and a transcription factor, right? And so we're lucky sometimes.
Mm.
Right, I mean, you know, Fleming didn't go looking for penicillin. Penicillin came and found him.
Well, for that matter, Newton wasn't looking for apples, right?
Do what?
Newton wasn't looking for apples.
Yeah, he wasn't looking for apples. He was a microbiologist after all.
I'm not sure if you're asking a specific question, but one of the important points of interest, in my opinion, is that this is very common in other sciences, but in biology and to a less extent chemistry is lacking, I think. And it is the ability to, it's a complex systems issue, it's the ability to integrate data from across fields, across scales, and across time, so that you can put together things that do not seem related, but they are indeed related through complex chains of interaction. So I think that's a very important aspect, particularly in genetics and understanding the notion of variation that you were raising. So how can we come up with a framework in which is in some sense universal, in the sense that any relationships that might exist become evident and are revealed outside the specific context in which they may arise. So I think that's a very important problem to solve, and one that I think might yield some interesting dividends. So integrating across scales in space and time is extremely important, I think.
Yeah, I fully agree. My argument is the current benchwork technology are based on a reductionist. Simplify everything to find one key element, control everything. But in the real world, it's not the case, right? Particularly dealing with genomics. Every time you have data set, I see hundreds of mutations. How can I simplify the system, find the one I believe is the major player, but in fact it may not be? I can make the story nice Western, right? Change. It doesn't mean it is the cause or the consequence.
Yeah, but that's just the point of complex systems. Complex
Right.
systems research takes the opposite view. It takes the sense that I learn more from a complex model of a very simple phenomenon that I do from a very simple explanation to a very complex phenomenon. So it's the opposite exactly. So this is a very different approach in complex systems than it is in reductionism.
Yeah, how to combine the multi-layered rich data with the reduction theory? That's obviously a very big challenge. I don't see there's any powerful solution right now. People go opposite direction, try to combine. It's not on the solid base. That's my feeling. Yeah.
Well, one of the points in complex systems is that things get it. I mean, the human mind can only process. We are essentially sequential processors when it comes to, abstract thinking. We can only think of 1, 2, 3 things at a time in our heads, and there are psychological rules that have been well researched and tell you the limitations. Whereas in the phenomena that we're interested in here, we have hundreds of things, thousands of things going all at once. So as you know, we may not trust the computer entirely, and we shouldn't, but computers will have the ability to handle all this parallelism and tell us something interesting that we may not have been able to see through there are limitations to the kind of analysis we can do. And I don't think we'll be able to continue without any more powerful device that will allow us to handle all that parallelism all at once. That we focus on specific regulators and enhancers and so forth. But is the dynamics of the things coming together that control or determine most of the outcomes, not just 1 or 2 or 3 at a time.
Yeah, that's what we can do right now. I hope there's some revolution happens to break this barrier between theoretical and the practical system and become universal approach. Yeah. No more? No more comments? All right, thanks.
Well, thank you for your attention today. As I said, I can use a little bit more of time. I hope to finish within 30 minutes. So this is unlike some of the previous talks in the sense that we're going to be doing some bioinformatics and computational stuff rather than applications and RNA transcriptomics. Although the results that you see presented here are applicable to a wide range of problems in both DNA and RNA. So life is wonderful, and the enormous variety of life is just amazing. It's extremely complex, as we were saying this morning. And we have learned in the last 30 or 40 years that molecular analysis is absolutely critical. And what we want to do is address this problem that we were discussing this morning of integration of data across spatial and temporal scales, very large. Not only in terms of nano, meso, and macro levels, but also in terms of integration across time. So this has been accomplished. Luckily, I can use an analogy to explain what is going to happen, because something similar happened early on with what today we call GPS. I guess the story here is that 400 years ago in physics, people wanted to know how planets move, and they would collect data of several points, astronomers did, and the question was, how do the planets move? And of course, Newton figured out a way through gravitation to understand that 3 points allow you to determine the trajectory of a planet because it moves in an elliptic orbit. And so it was necessary to establish a coordinate system that allowed the determination of this trajectory in that reference frame and the minimum amount of information necessary to determine the orbit. So today, jumping forward 400 years, we have GPSs that we use in our cell phones every day. So the idea then there is to provide a reference frame to locate any event in space and in time, and GPS offers exactly that about planet Earth. So what we want to do is something similar, but in a much more complicated sphere, biosphere, that is the sphere of life in all this diversity and extension. So here's the talk. Is going to be 3 pieces. The first 2 are going to be very brief because they are very technical, and I don't want to burden you with the technicalities. But at least they'll give you a sense of the research that is at the foundation of the system. And then I will explain how the system works briefly, and finally provide a couple of applications that may illustrate to you how it could be used. So I work in a field called DNA computing, molecular computing. The purpose of which is to essentially build computers out of DNA molecules. So the conventional wisdom about computers is that they're made of electronics. But this time we want to use real DNA molecules and the molecules to build entities that process information the way ordinary computers do. So this was a field initiated about 20 plus years ago by Adelman of RSA fame, the cryptographic scheme. And this year we celebrate the 21st version of the conference where experts meet to discuss these issues. So the basic idea is that the native properties of DNA can be used to self-assemble computers. And I'm not going to go into the details of that, but here's the smoking gun that shows that such things are possible. They were developed by Winfrey at Caltech about 15 years ago or so. So if we want to use DNA molecules to process information, then we need to encode that information in DNA. And to encode that information, we have to convince or twist the arm of DNA to do things that it was not supposed to do natively, that is process information. So we have to select DNA very carefully to make sure that it doesn't do what it's supposed to do natively, but it does what we want it to do in terms of processing information. So this is known as the code word design problem. And the basic idea then is to select oligos of the sizes varying between 10 mers or very short n mers to 100, 150, 200 mers that will allow you to encode that information, let's say on a chip, on a DNA chip. So what we want to, if you select those probes, we're going to call probes what is affixed on the chip, are we going to call targets what is put on the chip, and what is to be processed by the chip to obtain some information. So what we want to do is obtain what is called, what I'm going to call DNA NXH bases. NXH stands for non-cross-hybridizing oligos. So these are oligons that have been very carefully selected so that they don't interact with one another, on the one hand. But more important than that, they provide full coverage of the entire DNA space. What I call a DNA space is, for a given size of resolution, the length of the n-mers in the probes, we're going to select a complete set of, let's say, for example, n is 20, so 20 mers. We're going to make a selection of 20 mers in such a way that every other 20 mer, first of all, they don't interact with one another through cross-hybridization. So in particular, none of them are complementary to any other. Second, they don't even have enough affinity to form a stable bond. And third, any other 20 mer is going to stick to exactly one of those 20 mers. So it's going to, in other words, it's a universal coverage in the sense that if I drop any 20 mer in there, or something that has an overlap of 20 mers, is going to give me a signal of some sort to one of those probes. So that's what we're going to call a basis. And as I said, they have universal coverage. So this problem, if you look at it from the computational point of view, is very difficult to, is equivalent to optimization problem, the trivial assessment problem, which is known to be very hard to solve on conventional computers. So, however, what we have done here as the basic research is to find a method to select these probes very carefully so that the corresponding chip will have the properties that I just described of complete coverage and universality. So that, in other words, we have a solution to the code word design problem, which is very difficult conventionally. And we have done so here's an example of one such chip. I'm going to be presenting slides of this sort. White means non-cross-hybridizing, no signal, no attachment, whereas dark means full attachment. So this is a slide that shows is all against the other. So every probe, oops, excuse me, every probe is checked against the others, and the fact that it's basically white indicates that there's no cross-hybridization. So Gibbs energies have been normalized to 0 and 1, where 0 indicates no cross-hybridization. Minus 6 kilocalories per mole has been mapped to 1/2, and the minimum Gibbs energy below minus 6 kilocalories per mole indicates hybridization, stable hybridization. So as you see, most of them are non-cross-hybridizing, although there may be this one here may be hybridizing to some of them. So these are not, these are good because they're mostly white. If you take a random sample of the 20 mers, you're going to get something that is basically black. And so these are pretty good quality, although they're not the best quality we could provide. So once you have that basis, then I'm going to talk about DNA chips, although this is totally independent of that because most of us are familiar with microarrays, so they'll be easier to understand. So once you have your probes, then you fix them on a chip. In this case, we did an example of with 7 human diseases here. And that's 2, 4, 6, 7. We extracted from that a non-cross-hybridizing subset and obtain, and we run the genes associated with those diseases, and they produce the signal that you see here. So in other words, to obtain a signature for a particular genome or gene, what you do is slice up the genes by ultrasound or something like that, or cleavage, into oligos or fragments of size roughly the same size as the probes. And then you create the appropriate chemical conditions so that they hybridize, they attach to the probes, and they provide a signal by the intensity of the probe. So here, one of these, for example, the lighter ones reveal a high signal, and the dark ones are a low signal. And so you see this an example of if you were to take all the possible fragments in all the genes together, and you're trying to differentiate the diseases, then you would see this kind of thing. So this is an example of the signals that we would obtain if we didn't use a carefully designed chip. As you can see, there, two of them are very hard to distinguish, for example. And there's a lot more probes up here. This is 121 or so probes, whereas there's only about 49 probes down here. And the point here is, these are some analysis of the signal-to-noise ratio. And the point is that with fewer probes that have been carefully selected to avoid cross-hybridization, you get much cleaner and lighter signals. And we all know that microarrays are very sensitive to there's a lot of signal, and the signal-to-noise ratio is very low. So the first lesson that we learned from this system is that larger is not necessarily better, that putting the largest amount of probes on the chip is going to create a lot of noise, and here's a way to clean out the noise and provide better signals for what you're getting. So that's an example of a careful design, how a careful design provides you a lot more information, because it's much easier to distinguish two diseases here by this signature on that chip, on the clean chip, on the non-cross-hybridizing chip, than you would get on an ordinary DNA chip or a microarray up here. So that's the first part. The second part is that we have found a much better way to design these chips so that essentially give universal coverage. This example here was done only for a peculiar design for those diseases. In this case now, we have been able to come up with a design that allows you, that gives you a truly universal chip that will provide a signature for anything, for any kind of set of oligos that you throw on that chip will provide a signature of some sort. And I don't want to go into the technicalities of this, but you might ask, how does this relate to real life stuff? And the upshot of it is that on this chip, the decision that this chip makes about a hybridization event agrees with what it would happen in real life as compared to the model, with a probability of 80%. In other words, this is not a perfect model, but it gives you about 80% of the truth that you will see. And of course, there's a trade-off. The model is a little bit off, but on the other hand, it affords some advantages that you wouldn't have otherwise. So these are the technicalities of the design of the chip, but to give you an intuitive idea of what's happening here, it boils down to packing. Designing a universal chip is a problem in sphere packing. All of us have packed our suitcases, and we know the problem, right? We know that if I put something here, I cannot put something else there. So and the man in the street actually has a pretty good idea how to do this packing. This man, as you can see, this man, what is he doing? He's trying to sell his fruit, right? And he's trying to maximize his exposure, right? So what does he want to do? He wants to pack the maximum amount of lemons and oranges and so forth into the minimum amount of space. And that's exactly the problem that we need to solve. The only difference is that that problem has to be solved in a very high-dimensional space, not just three-dimensional space. But if you have 6 MERS, you have to solve the problem in 24-dimensional space. If you have 20 MERS, you would have to solve the problem in 80-dimensional space. And so here are examples. So this is a nice packing of pennies in a 2-dimensional space. That gives you an idea of the optimal solution. It's a hexagon. Believe it or not, it took 200 years for mathematicians to prove that that's the optimal solution. This is an example of cannonballs that are the optimal solution in 3-dimensional space. And this is an example of some phenomenon that illustrates the packing in 4-dimensional space, which would correspond to just simple ACGT. So we are trading off this optimality for reasoning in high-dimensional space. So that's just to give you an idea of what is involved. So fortunately, people have been working for the last 200 years on this problem, last 200-300 years on this problem, and they have found very interesting solutions. The ones I'm going to use today, to show you today, have to do with this so-called Leach lattice. So anybody who works in physics or even chemistry will know about the Leach lattice, because it has to do with crystallography and so forth. So this is a surprisingly nice packing that happens in 24-dimensional space that gives us a nice universal chip for 6 MERS, for example. So that's just to, so this was developed back in the 60s and 70s to solve problems in communication theory, but we have been able to apply that to the design of the DNA chips here. So here are some statistics on the best known table that shows you the best known designs for 6 MERS, 8 MERS, 12 MERS, 16 MERS, 20 MERS. So we have developed some procedures of evolution in vitro to obtain some chips, but they're not as good as the ones that I'm presenting today. So now that's the second part. Let's move on to the application part. How am I doing? I'm doing good. How much left? Oh, great. Great. So let's see if we can talk a little bit more about the applications, because that's the juicy part that you probably want to see. So what is this chip good for? So I'm going to give you 2 applications that we're developing. And one of them has been fairly developed. There was a PhD student 2-3 years ago, and the other one is the project. One of the major projects that we have today is the Tree of Life. And so I'll explain them in turn. So what is it that you want? I mean, this is the gadget. What is it that you want to do with it? Well, you want to extract information out of DNA molecules, right? In one way or another, either expression levels or correlation between expression levels, genomic analysis, transcriptomic analysis, and so forth. That's what you want to do with it. And so to give 2 proofs of concept how it is possible with this kind of gadget, I'm going to talk to you about semantic analysis. So some of you may have come across this. All of you have done semantic analysis in some form or another in using your Illumina, Pacific Bio, etc. It's exactly what you want to do. You want to do data mining. You know, you can get all this data, and you go through all this trouble in the lab to get all this data. But if you can't analyze the data and get the conclusions that you want about your problems, all this is for nil. So semantic analysis has to do with that. So the idea of semantic analysis is that DNA can process information in a semantic way, in a way that you and I would do it. So semantic has to do with the idea that I don't really care how you represent the information in the computer. I care about what it means, or what conclusions I can get in human terms, in terms that you and I can understand and use. So in other words, I don't want data. I want information that I can use. So Baum demonstrated back in the early days of biomolecular computing that this is indeed possible. So he said, well, here's my database. It's a bunch of DNA molecules like my chip, like the chip that I just told you. It doesn't have to be on a chip. It could be in solution, because the molecules are not cross-hybridizing, so you can just toss them into the tube. And so this is a database of a company, for example, with all their customers. And each DNA strand encodes something like the customer's name, an ID, and balance, and whatever else. And you want to know John Smith. John Smith's record, how much does he owe, what is his account number, blah, blah. So what you do is drop a probe, like a fishing line, into that, and that contains the complementary strand to the one that contains the record for John Smith. And of course, there will be, if this complementary is going to be hybridization, and the fact that they're non-cross-hybridizing will assure you that only that particular record for John Smith will be pulled out of this mess here. Of course, there could be billions of molecules in there encoding billions of customers' information, but only the one that is relevant to John Smith in some sense or another, because there's some affinity in that complement, will be pulled out. So then there you go. That's how semantic memories work. And they're semantic because although DNA is encoding information, it's not encoding information the way conventional computers encode it, that you have to have an exact match to be able to retrieve that based on an address on memory somewhere. The molecules are moving around and they contain the information. They're not pinned to a single address. They're just looking for the complement. And as long as the complement that presumably matches the information that is being requested, you're going to get only the right thing and the right thing, if it is there, of course. So that shows that semantic memory, that processes information the way we humans would, is possible to retrieve from DNA devices. So the best known processor of semantic information in electronics is called LSA. So LSA stands for latent semantic analysis. There's variations of it. It's an example of so-called single value decomposition and principal component analysis, if you wish. And that's what it is based on, but this was originally done for electronics. So here's an example of a particular semantic problem to make the point. It's called textual entailment. So what is it? What is the problem? The problem is you are given two texts, the T for the text and this H for the hypothesis. And the text essentially gives you a situation or a context, and the hypothesis gives you essentially a question. Is this true or not in that context? So in this case, the shooters escaped as other soldiers tried to give aid to the wounded. That's the situation, that's the text. And now you want to know whether this hypothesis is true based on that text. And the hypothesis is the gunman escaped as other soldiers tried to help the wounded. So if you give this to an ordinary computer, the computer is going to be befuddled by the fact that shooters and gunmen are different words. They are spelled differently, so they're considered to be different words by the computer. But you and I know that in this situation, shooters and gunmen are equivalent, right? They're the same thing in the context. So you and I will say, sure, the hypothesis follows from the text. But the conventional computer will say, gee, I'm not sure, because those two words are different. It doesn't have a sense of the semantic equivalence of the two. So it's important in that case, for example, to solve this problem of word disambiguation. How can the same word can have two different meanings, like the word belt here? And it's in this case, you need to, semantic analysis is important to distinguish between the two. And then what my student did was to run a study using the DNA chips of the sort that I have described to you, and then using a word net, which is a network developed at Princeton by Miller in '85, to try to capture the idea of word knowledge. Because one of the things that we use in an inference, like solving the entailment problem, is we pull every bit of knowledge that we have in our head about gunmen and shooters to solve the problem, right? To decide on the equivalence. So by encoding that word net into DNA molecules, then here's the upshot of the results. So we were able to, by a very simple method of DNA hybridization, we were able to at least be competitive, if not better, than the best methods for textual entailment. This is a standard problem in semantic analysis, and there's a yearly competition where entrants use their solutions to solve a standard set of 800 questions of the type that I gave you. And there's winners and losers and all that. But here are the results that they got versus our results. And they're very competitive. So these are very sophisticated methods with a lot of computational intelligence and things like that. But we were able to match that performance based on simple DNA hybridization on this chip. So that gives you a sense how you can perform semantic analysis on these DNA chips using biochemical ideas. So the next example is the last example is the tree of life. This is some chart here about the power of along 3 axes in terms of speed, size, and semantic quality of a number of semantic analyzers. So I'm not going to go into that. The last part then is the tree of life, and so this is closer to home to most of you. So what is it that you want to do? Well, we have fossils and we have DNA, and DNA is a recorded history of evolution, that what's happened in the past to be what we are today. And what is it that you want to know? Well, given your genome, or the genome of a number of species, you want to decide. A phylogenetic analysis will give you some idea of the evolutionary paths that those organisms have followed through their DNA record to become what they are today. So a standard project in biology, in evolutionary biology, is to build a tree of life that gives us a sense of how life has evolved on planet Earth. So what we're doing is, the second proof of concept I want to show you is show how this kind of analysis is useful to build to do phylogenetics and evolutionary analysis. So so initially we took 6 bacteria here on one of the good DNA chips that we designed, and then what we wanted to do was test the hypothesis that we can use the signatures that those genomes produce on these chips to build a phylogenetic tree for those organisms de novo, absolutely from scratch, in a way that is perfectly consistent with what biologists have elucidated in the course of 30 or 40 years of research to build more or less the same tree. And so that was exactly, so what you are looking at now is the signatures of the 6 bacteria on one of the chips that we have designed. And then here are the bacteria that we used, and this is more or less the size of the genome as downloaded from gene banks. And then we used only the open reading frames for that. And then we used, these are details on the chip that we got, but the upshot is this is the phylogenetic tree that biologists have developed over the course of years, and this is the phylogenetic tree that we obtained using the chip. And as you can see, it's essentially producing the same phylogenetic tree except for a small variation here that these 2 are swapped. And even that is debatable within this tree, whether that should be correct or not. So the point of all this then is that indeed the amount of information that you obtained essentially instantly from this chip is equivalent to in content to the information that you can obtain by other means from standard biological methods. So we repeated that's the upshot of that. We repeated the experiment with 17 bacteria, and we essentially got the same results. So we reproduced de novo the same analysis that biologists have obtained in by other methods, much more costly in terms of morphogenetics and so forth. So the tree of life that we are developing now, here's my last slide basically, or next to last slide. So what we are engaged now is in the tree of life project, which is trying to build an atlas of life based exclusively on the signatures that an arbitrary genome produces on these chips. And here's an example. If you run the most elementary chip, is the one that has two values, the all A's and all G's, for example, and the signal that you'll get from there is the GC content of the organism, which is the simplest, most primitive kind of chip that you could get. On a more sophisticated chip, for example, here's an example. We can get the stop codons. You just look at the stop codons for a chip. These are the codons. And so we took 12 organisms representing 12 an exhaustive representation of the coarsest level of all life. And it produces these signals on that chip, which is still a very, very rough, very primitive chip. But even there you can see that this first probe, there are 12 probes, I'm sorry, 3 probes, these 3, and you produce the signals here. And then even that probe, that first probe, is an excellent marker to distinguish this organism from all the others. And so forth. So the fact that if you take a vector of the 3 signals coming from the 3 probes, you'll see that that's a complete set to distinguish amply each of the organisms from all the others. So it gives you this is a very coarse chip. It's a very small, only 3 probes, and gives you that differentiation already. So the plan now is to enrich the chip and get a more sophisticated chip. And if all goes well, we'll be able to, at least in the case of the bacteria, we were able to differentiate the chips down to the order. And I think with a richer chip, we'll be able to differentiate down to the single organism, single species, and even maybe single organism if the chip is refined enough. So this is just beginning now. This method is absolutely general. If you want to use the chip to distinguish, it's a universal chip. So if you just take a single gene or a single segment of a gene, it will give you a signal of some sort. And the point is that on the same chip you can compare everything. Everything, absolutely any DNA will give you a signal, and it's all on the common reference frame. So in other words, it doesn't matter if it came from a gene or a completely different chromosome or organism, it'll give you a signal of some sort that you can compare on the same frame of reference. So perhaps then we'll be able to determine the trajectories of life, as Newton was able to determine the trajectories of the planets. So, thank you.
I do believe over the time, so we got a couple of minutes. We need to give away the certificates. We'll take one question, okay? One question only. Okay, and then we got to do certificates.
Have you tried to use all your modern species of bacteria? Have you tried the high animals like primates or even human sequence to prove it?
Yeah, we have actually this is a study that we're doing on housekeeping genes. You know how this differentiation is being made only on the housekeeping genes for those organisms. But of course you can try it on mitochondrial DNA, or you can try it on genomic DNA, or coding DNA, non-coding DNA, and so forth. So there's an unlimited number of things that you can do here.
Yeah, but like human genome is such big size compared with bacteria, and then the specificity will decrease, or you need to increase the combination?
The specificity is the same. I mean, it depends on the chip. If you get 6 MERS, you get certain specificity. If you get 12 MERS, you get more specificity. 18 MERS, even better. So it's like a zoomable camera. The longer the probes that you get, the more specificity you will get.
Can you expand the concept to create individual identity chip based on, for example, SNP information?
That sounds like a long explanation, maybe for a minute. We should take it offline. But I'll be very happy to talk to you about the details.
Thank you very much for this opportunity to give me presenting my recent translational research, actually, because the rest of my job is diagnostic one, and I enjoyed your recent talk about the disease-causing mutation, which is a big issue now. This one is about the transcription factors. Today morning, there was a discussion in this regard in breast cancer, and mentioning breast cancer stem cells and its possible grand and great effect on metastasis. SLUG and SOX9, as you know, both are transcription factor, major transcription factors, doing something important in breast cancer. And I present they cooperate together to regulate the good microenvironment and niche for breast cancer to be metastasized, actually. Metastasis is the major cause of cancer mortality. Metastasis is a multi-stage process, and we can consider a pathway, molecular pathway for that. And I would like to name it the metastatic molecular pathway. Successful initiation of metastatic growth, metastatic colonization, accomplished only by a minority of cancer cells that reach distant size. A small population of cancer stem cell is critical for metastatic colonization, and as you may remember, the seed and soil or metastatic tropism is the main dogma for this one. And cancer stem cell has some very well-known characteristic, including from self-renewal to apoptosis resistance. And about epithelial to mesenchymal transition, there is an emerging concept for metastasis in this regard, and it is the thing you can see in embryonic morphogenesis, in adult wound healing, and in metastasis. Wherever we have migration, invasiveness, apoptosis resistance, and these are all the characteristics of metastatic cells. EMT transcription factors are somehow known, including slug, snail, twist1, twist2, and the signaling pathway, which are already known, well known, are TGF-beta and WNT-beta-catenin are in this category. There was a very good paper published in 2012 by Weinberg Group in New York. It says SLUG and SOX9 cooperatively determine the memory stem cell state as master transcription factors. SLUG and SOX9 master transcription factor, to have a very quick review on that, actions of key transcription regulators is sufficient to convert differentiated epithelial cells into long-term repopulation epithelial stem cells. And as autoregulatory program, which is activated by SLUG and SOX9, involved in inducing and sustaining the stem cells and stemness. Adult stem cells are cancer stem cells similar to embryonic stem cells, maintain their stemness via master regulator mediated autoregulatory networks. And it was really nice for me at the end of the discussion. I will ask to go back and address today morning discussion and see the expression pattern of these proteins, very limited number of protein transcription factors in the study of proposed study by you, Doctor. Actually, the patient, it is well known and it is shown, confirmed that patients with primary tumors expressing high levels of both SLUG and SOX9 had a significantly lower overall survival rate compared to those non-expressed, this one, in high level. Metastatic niche includes fertile microenvironment, supports stem cell maintenance and manage cell function and proliferation, including cell vascular network, soluble factor, nutrient, metabolic components, and ECM, as well as VEGF, membrane metalloproteases, LOX, and something like that. ECM and metastasis also address regulatory signals, sophisticated organization, and these two proteins, periostin and tennessine, as metastatic niche component for cancer stem cell. Why? Because in 2012, again in Nature, there was this published paper, Interactions between Cancer Stem Cells and Their Niche Govern Metastatic Colonization. Again, in 2011, in Nature Medicine, it is published that breast cancer cell produce tennessine C as a metastatic niche component to colonize the lungs. Okay, these two proteins, not transcription factors, periostin and tennessine, are also important as metastatic niche, in parallel to those SOX9 and SLUG. And they are studied at the same time, roughly in 2011-12, and published by different scientists and in different groups. We try to link between cancer stem cell and metastatic niche, addressing these two, and published this article in 2013. There is a crosstalk between breast cancer stem cell and metastatic niche as an emerging molecular metastatic pathway in tumor biology, and it is the thing that we hypothesize. As you can see, SLUG and SOX9 is at the top, and there are some intermediate components, then linking to tennessine and periostin in normoxic and hypoxic area. And you can see the relationship they have with EMT and hypoxia and HIF1. In the same year, a paper published in Nature Cell Biology saying that perivascular niche regulate breast tumor dormancy. And it addresses periostin particularly, and tennessine also in the same pathway of TGF beta 1. In response to that, and considering our previous studies, we published an answer that perivascular niche governs an autoregulatory network to support breast cancer metastasis. The hypothesis was simply overexpression of SLUG and SOX9 transcription factors may increase the expression of periostin and tennessine genes, and vice versa. Their knockdown may significantly under-regulate and lower expression of periostin and tennessine. I need to go faster. This study was done by my student, master student, and he recruited the constructs, including SOX9 and slug, as well as silent RNA for slug and SOX9, and just transfected the cells via lentivirus, and then did real-time PCR in MCF7 and MDA MB231 cells to compare these two invasive and non-invasive breast cancer cell lines. As you can see, in normal MDA and MCF7, there are significant different pattern of expression for slug, SOX9, periostin, and tenecin. It is the thing we've done in the lab by real-time PCR, and you can see MCF7, which is resistance to invasiveness and metastasis, showed a really lower, significantly lower expression of these 4 components. Talking about upregulation of the genes and transcription factors, the overexpression of slug and SOX9 significantly upregulated the expression of periostin and tenecin, which SOX9 is more obvious, and but as it is shown, their co-expression and cooperatively expression of these 2 transcription factor is much obvious, and it may propose that their co-expression and cooperation is significantly more powerful than single transfixion and overexpression. As you can see, there is about 1,000 times for upregulation of, in particular, periostin, and then about 50 times of tenecin for when they are co-expressed, compared to just single overexpression. Consistently, in knockdown slug and SOX9 downregulate periostin and tenecin. In this experiment, we've done in MDA cells, because it, as you could see, the expression levels of these components were high in MDA invasive cells, breast cancer cell line, and then we knocked down MDA 231, knocked down these slug and SOX9 in MDA 231, and quantitatively measured the gene expression of periostin and tenecin gene expressions. And as you can see, significantly, both of them are decreased, and again, periostin is more obvious and much more significant. To discuss, slug and SOX9 expression may have synergistic effect. Slug and SOX9 co-expression is required for periostin and tenecin utmost expression. Periostin is likely one of the most important factor, even much more than tenecin, regulated by slug and SOX9. And as acknowledgement, I need to address my master student, which is now a PhD student in Spain, and the grant that it was funded by the university in Iran. Thank you very much.
All right, do we have any questions, folks? Anybody have any questions, comments? No, sir, nothing?
Thank you.
Thank you.
Yesterday we have lost Dr. APJ Abdul Kalam, former President of India. He was known as Missile Man of India and a great eminent scientist. So today I dedicate my presentation to the great soul. Well, I, Saurabh Chaudhary, research scholar from Guru Gobind Singh Indraprastha University, presenting my work entitled De Novo Transcriptome Assembly and Identification of Cold and Freeze Responsive Genes in C. buckthorn. Well, first of all, I would like to introduce my subject plant, that is C. buckthorn. It's a hardy deciduous shrub, belongs to the family Allignaceae, bearing yellow to orange berries, which have tremendous medicinal nutritional values. Indian Himalayas is the second richest source in the world of C. buckthorn. Beside medicinal and nutritional value, it has ecological importance also, prevents soil erosion and helps in land reclamations. Besides this, it grows in extreme environments, so it's maybe important source for new genes alleles for abiotic stress tolerance. To keep this in mind, I have been assigned with 3 objectives. One is C. buckthorn transcriptome assembly and analysis using NGS, deep stage-based identification of differentially expressed gene during cold and freeze stress, validation of selected cold and freeze responsive gene using QRT-PCR. Well, for transcriptome sequencing, development of C. buckthorn seedlings. We developed C. buckthorn seedlings in our laboratory. Seeds were surface sterilized and germinated hydroponically at room temperature. And after 3 days of germination, seedlings were transferred in growth chamber with 28 degrees Celsius relative humidity and 55% and photo period of 16 hours light and 8 hours dark. The second step is total RNA isolation and quality control. Total RNA isolated using modified CTAB method, which was modified in our laboratory, standardized in our laboratory in 2009, because C. buckthorn contains high secondary metabolites and bioactive compounds. Therefore, it was really difficult for us to isolate the RNA, good quality RNA. So we standardized a method which is modified CTAB method. The good quality and high yield of total RNA was obtained from leaf and root tissues, and RIN value more than 8 were further processed for next generation sequencing. For the transcriptome assembly, high throughput next generation sequencing on Illumina Solexa platform was used, and it was outsourced. And we have received raw data, raw short reads, which was filtered out using NGS QC toolkit developed by Jain et al. In 2011. And this is a summary of the results, short reads which we obtained, that is total number of reads from leaf and root tissues, and high quality reads and the percentage of high quality reads. Approximately 94 million short reads were generated from the Illumina Solexa sequencing, and these were submitted to NCBI short read archive. Out of that, approximately 86 million reads of high quality were further taken as for assembly. Now, the biggest challenge against us was how to assemble these huge amount of data, huge amount of short reads. So we used different assemblers, and for the optimization of transcriptome assembly, including Velvet, Oasis, Abyss, SOAP De Novo, CLC Genomics Workbench, and Trinity. Well, for short read assemblers that perform assembly as a function of k-mer, lower the k-mer, higher is the computer's RAM requirement. Thus, for RAM intensive assembly tools, high quality reads obtained from Illumina platform were fed to CD HIT 454 suite to remove redundancy, and obtained about 24 million non-redundant short reads. So we analyze both non-redundant as well as redundant short reads with this, and in the first approach we compare de novo assembly as a function of best k-mer. In that case there are total number of contigs shown by these bold bars, and average read length and N50 read lengths. On the basis of these, we found that OSIS and ABIS be most appropriate tool for assembly in this case. But as we proceed to second approach, that is comparison of assembly as a function of additive k-mer, then we have found the similar results that ABIS and non-redundant ABIS, OSIS and non-redundant OSIS was to be best suited. Then we use another approach in which we compare de novo assembly as a function of additive k-mer followed by TGICL suit. TGICL suit is a is a suit in which long reads were taken for that, so that we use additive k-mers in TGICL suit. In this, it's a two-step strategy, and we assemble the long reads. In this, you can see that again ABIS and non-redundant ABIS, OSIS and non-redundant OSIS is found to be the most suited, with the average read length and N50 read lengths increases significantly in these cases when we use this additive k-mer approach followed by TGICL suit. So after this, we compare the de novo assembly as a function of ability of short read to map back onto the Seebeckton transcriptome. In this, we map back all the unigenes, all received from the different assemblers to the Seebeckton transcriptome, and we found 90% hit with the existing transcriptome, except in case of CLC Genomics and Trinity. These are the results of various assembly parameters. In case of ABIS, we obtained 88,297 unigenes, and in case of OSIS, 86,124 unigenes. Well, this also N50 and average read length also increases significantly in these two cases. Now, these are the summary of my result. On the basis of various assembly parameters, ABIS was found to be the most promising assembly software, which resulted into 88,297 putative unigenes in Seebeckton. BLAST-X algorithm was to annotate Seebeckton transcriptome. In this 41340 had significantly the existent non-redundant protein database of NCBI. Higher sequence homology was found with Vitis vinifera, followed by Racinus and Populus. BLAST-2GEO suite was used to assign gene ontology terms, and functional annotation revealed that 3,035 unigenes to be involved in different stages in Seebeckton transcripts. Many transcription factor and SSR motifs present in the transcriptome also identified. These are the top hit sequence similarity, with Vitis vinifera is the most one. Gene ontology classification, biological process. This is on cellular component basis, and then molecular function. The work is published in PLOS in 2013, optimization of de novo short-read assembly of Seebeckton transcriptome. Now, for my second objective, that is identification of cold and freeze responsive genes, we used deep SAGE, a combination of classical SAGE and next generation sequencing used for global gene expression analysis under certain environment. It is also known as digital gene expression profiling. For digital gene expression profiling, again, Seebeckton seedlings were grown, and in that case, different cold and freeze temperature were given to the seedlings, that is at 4 degree, and freeze treatment at -10 degree. Now, again, total RNA was isolated, and the 17 base pair tag were generated as per the manufacturer protocol. In case of digital gene expression analysis, tags were directly sequenced using NGS, Illumina Alexa platform, and analysis were done, which results into a large number of differentially expressed genes in response to cold and freeze stress. Now, differentially expressed genes were annotated by sequence similarity BLAST with our existing Seebeckton transcriptome, and around 450 genes identified responsive to cold and freeze stress in Seebeckton. This is the pipeline for bioinformatics analysis of raw sequence reads in DeepSage, where raw sequence data will converted into clean text, then gene annotation and normalization, and then screening of differentially expressed gene was performed. This is a distribution of total clean text and distinct clean text received in DGE sequencing. Now, the result of differentially expressed genes, normal versus cold, we have identified 2051 genes upregulated and 1821 downregulated. In case of normal versus freeze test, 2278 were upregulated and 1774 were downregulated. We also compared cold and freeze stress conditions, and we found 2210 upregulated and 1788 downregulated genes. Gene ontology and KEC pathway analysis were also performed for differentially expressed gene. Geoterms suggested metabolic, cellular, primary metabolic, cellular metabolic, and macromolecule metabolic process genes were found to be abundant under cold and freeze stress. 28% and 15% of differentially expressed genes were characterized as gene responsive to stimulus and stress, respectively. Photosynthetic, carotenoid biosynthesis, and plant hormone signal transduction, vitamin B6 metabolism pathways was found to be most significantly enriched pathway during cold and freeze stress in Seabuckthorn. Now, the result of DeepSage were validated by qRT-PCR. The differentially expressed gene blast with annotated unigenes of Seabuckthorn. 22 genes from DeepSage data were selected, involved in cold and freeze stress for validation. The Seabuckthorn seedlings were again given the cold and freeze stress for real-time PCR expression. These are the expression of all the 22 genes which used in qRT-PCR expression analysis, MAP2K, ADC, PP2C, AP2ERF transcription factor, RPL50, ATP19, SOR, HSP70, WD40, KC, and PEP. And these are the fold changes. Some are upregulated and few are downregulated. This is the expression profiling of genes under using qRT-PCR. And the work is published in PLOS ONE this year only. So from the study, we have concluded the standardized the high quality RNA isolation from Seabuckthorn and established a Seabuckthorn seedling in laboratory. World Transcriptome data obtained using NGS platform provides an upper age as reference data sets for further studies. Unigenes with no relevant hit in NCBI, non-redundant protein database, were considered to be Seabuckthorn specific and can further be characterized in future. Standardization of assembly tool for NGS data has further in analysis of NGS data for other species. We also identify 7421 putative Seabuckthorn transcription factor genes, developed USMM unigene specific microsatellite markers for Seabuckthorn. Many abiotic stress responsive genes, especially for cold and freeze tolerant in Seabuckthorn, have been identified. Research leads, genes identified to be involved in cold tolerance need to be further characterized. Some of these be explored for development of cold tolerant transgenics. Assembler ABIS optimized for NGS assembly can be used for further similar studies. And in the last, I would like to thank Department of Biotechnology, Government of India to provide the fund for this project, ITS, DST, Government of India for providing me travel grant, and my university, Guru Gobind Singh Pasta University, and of course, Omics International Conference to represent my work here. Thank you.
All right, do we have any questions? Any comments or questions? Okay. Yeah.
It was a nice talk. Could you please tell us about the medicinal importance of this plant?
Yeah, actually, there are a number of therapeutic and medicinal uses of this plant, including it is used as a UV protectant in anti-cancerous. It has anti-cancerous property also, and there are lots of more pharmacological importance has developed, like The main is anti-cancer. It is contains antioxidants, number of antioxidants it contains, and every part of sea buckthorn, like leaves, fruit, stems, and roots, every part of sea buckthorn is used in medicinal and nutrition.
Is it available in the market?
Yes, yes, it is available in the market. Where, the DRDO lab India has first discovered is juice, sea buckthorn juice, known as Leh berry juice. DRDO lab situated in Leh, Ladakh, and colleagues from my colleagues from China and Russia, they must knowing this plant very well, because it grows abundantly in Russia, China, and of course in the Indian Himalayas region.
Thank you.
Yeah. Okay. Thank you.